Ingrid, you're muted.
Hello, everybody. Welcome back. After the first successful day yesterday, we will again have a number of interesting lectures and tutorials today. Our first lecture will be on multi-scale visualization. This is a very relevant topic in visualization in general, but especially in biomedical visualization, as we already learned yesterday in Jan and Noska's talk on data sources, or Niels' talk about omics data. So the lecture will be given by Helwig Hauser. Some of you might know him already or have heard of him. So Helwig Hauser is a professor in visualization and the leader of the Center for Data Science, CDAS, at the University of Bergen in Norway. So the center has a particular focus on interdisciplinary research and data science, but also on data science education and applications. Helwig joined University of Bergen in 2007, where he has built up a research group on visualization, which became one of the most well-known groups in visualization in the world. So before moving to Bergen, Helwig was the scientific director of the BioMedVis Research Center in Vienna, Austria, where he earlier also led the basic research group on visualization. So he got his education in computer science at the TU Vienna, where he also completed his PhD and habilitation. And with this, I'm happy to welcome Helwig here at the BioMedVis Spring School as a speaker. And we are all looking forward to your talk. And Helwig, please start. The stage is yours.
Thank you very much, Ingrid. Can you hear me?
Yes.
Excellent. Thank you, Ingrid, for this amazing introduction. Very kind words indeed. Thank you so much. First of all, maybe I can say I have so much respect for the organizers of this spring school for what they have pulled together here. It's a truly amazing program, and I'm excited to be part of that. I have to say that these lovely organizers, they gave me an impossible task to complete, to talk about the topic of multiscale biomedical visualization in just a short hour or so. So I hope you can bear with me. I can only scrape the surface of this topic. It's a highly fascinating topic. I'm not sure I'm the most competent person to talk about it, but I'm certainly very fascinated of the topic. And I will share a few of my impressions and thoughts with you today. And hopefully we can have some exchange about your questions and thoughts to this topic after the presentation. So with that, I just send you greetings from Bergen in Norway, where I am right now. And maybe one day you can come and visit, and either for the purpose of talking about visualization with us or just for the fun of hiking the wonderful environment here. So the plan for today is to basically introduce the topic a little bit, just to make sure we are on the same page somehow. And then I thought I want to basically go through a few different aspects of multi-scale biomedical visualization with you, giving you some selected examples. I really do not have the time to dive into the details here. It would be very interesting for some of these topics, but this has to wait for some other day and time, I guess. And hopefully there will be time to wrap up at the end with a few lessons learned. So let's just get started with the introduction. And maybe it's appropriate to first talk about terminology. And here probably the word scale and maybe scalability is the first that comes to mind. So a scale is first and foremost, it's a word that is used both in regular language and with certain specific meaning in maybe science and technology and other fields. And we find it in different contexts. So one is that we use it to indicate a measurement scale. We measure something maybe in centimeters or in seconds or maybe in other terms. So a scale establishes maybe an axis of reference for something that we want to talk about. Another use is that of an order of size So I think many of us have looked at this famous old movie or short movie, The Powers of 10, where you have an infinite zoom almost from the smallest scales to the largest scales, illustrating very nicely that our world is composed of many different scales at different sizes. And then sometimes when we take the word scale into use, we mean simply big or large amounts of data, maybe big problems. And that relates also to the notion of scalability. So I guess many of us have enjoyed reviews coming back after submitting a paper, and then there was a question, well, but does your technique also scale to larger data? So scalability would be then a property maybe of an algorithm or a solution or not also. So this could be seen as a reference. Here I've just included little slide deck of zooming into and out of the Mandelbrot set, which is a famous fractal. And it's one of the big ideas that has also fascinated me for a long time already. Actually, when I was a teenager, I got the book, The Fractal Nature, the Fractal Geometry of Nature by Mandelbrot, and it fascinated me so much that it never let me off the hook again. So it suggests that when you think about the world, biomedical world, but also the world beyond that, that there's a lot to see on different scales. And this leads then also to this notion of multiscale whatsoever, and it comes in a variety of domains. Not surprisingly, I mean, as many domains are concerned with the world, different aspects of the world, we find this multiscale aspect also in the different domains. Maybe most prevalent it is in the large scales. So then we talk maybe about astronomy. Here I've taken an example from Andrew Hansen's work, an early work about coming up with a system that allows you to explore the universe at a wide range of different scales. Here in the middle, you see a list of spatial scales, where in the middle here we have the human scale, 1 meter approximately. And then as we go up in the powers of 10, we get to larger and larger scales beyond the Milky Way to the whole universe maybe. And if we go the other way, we go into more details, and that is then the small scale, maybe the atomic or the molecular scale, where we have equally many, if not more, scales to the small sizes. If you have ever used mapping applications like Google Earth or the like, then this is also obviously an example where many scales are combined, both technically and in terms of visualization and representation. Today, the topic is, of course, mostly about biomedicine, and I will try to focus on that area in the remainder of this talk as much as possible. So in biomedicine, we clearly do have these scales, in particular when we include the bio into the consideration. Then we are not only interested in, say, the human scale or the organ scale, as it's often the first thing when we do maybe medicine or anatomy or the like, but then we really want to connect this all the way to the smallest scales where we have molecules and atoms. And that creates a wide range of scales, say from meters down to nanometers maybe, and from years, when we think about a whole life, maybe down to even picoseconds, nanoseconds, picoseconds, when we talk about interactions between atoms. And I think this really opens up some interesting questions. So in visualization, for a long time, we were challenged by visualizing anatomy, so I don't know, 20 years ago, 30 years ago, it was a big challenge how to accomplish 3D visualization of an entire organ. I think today we need to think much further and into the realm of physiology, where we add function to the structural aspects of medicine. And then it's unavoidable to dive into multiple scales, because this means that we also have to think about time, and there we have even more scales to consider between, say, an entire life and the interactions between atoms. So these are maybe two of the important scales that we have to think about when it comes to biomedicine. Of course, for us, it's always important to see, well, what are we visualizing? Are we visualizing data? Are we visualizing models? And as first, we may maybe think about data that we get from measurement processes, from imaging devices. And here, it's an interesting question. What about the different scales? And obviously, we do have medical imaging on the largest scales. There's a variety of scanners used in hospitals these days that give us pictures, 2D, 3D pictures of human anatomy, and that's a rich data source for visualization. On the other side, if we look at the small scales, we have the atomic, the molecular level, and there we have both the computational processes that, for example, are numerical simulation, as well as some highly advanced microscopy imaging techniques that give us data from these structures and processes. I found it quite interesting to follow an observation by Johnson and others, here illustrated from the Mesoscope project, where the suggestion was maybe we are good at acquiring data on the large and on the small scales, but maybe we are missing out a little on the so-called mesoscale. And I think this is something to think about, and also an opportunity for the future. So on the one hand, as I just mentioned, we think about data that we visualize. On the other hand, we need to also think about modeling and modeling of physiology. And if we dare consider multi-scale phenomena, then it really becomes a big challenge. Modeling an aspect on one scale is often a big challenge by itself. But modeling a system that is composed of multiple pieces that should play together on multiple different scales, that's a big challenge. Here I've taken just two pictures from multiscale modeling publication a couple of years back that illustrate this. So here we have, say, the wound healing process, and you can see that it happens on many different time scales, from small early phases to much slower long-term phases. And here's another picture that illustrates processes that are important when it comes to the question of whether or not you may get a stenosis in a vessel. And you can see that we have here multiple aspects of this system's picture that range from milliseconds to weeks, as well as from micrometers to centimeters. And they are playing into each other in a quite complex fashion. So setting up these multi-scale models is a big challenge. But what is maybe then even more a difficult question is, can we use these models for numerical simulation? And there it really becomes a difficult question how to have these models interact with each other. Usually it's not possible to run the numerical simulation on the smallest scales to also account for the processes that are described by the large scales model. So you need some way of interaction between these models and some exchange of states. And this is really quite a big challenge and difficult to achieve. So with this in mind, we could ask ourselves, what do we find in visualization related to both the data and model scenario when it comes to physiology over multiple scales? We did a survey a couple of years ago. Unfortunately, we didn't publish it, but we looked into what can we find in terms of visualization, and we tried to map it out also a little bit. And here you see a bit of a summary visualization of what we found. And it's quite interesting that if you look at the different spatial scales here we categorize them into molecular scale, organelle scale, cell scale, tissue scale, and organ scale, that there's a lot of visualization found on the organ level. I think that's influenced by the early works on anatomy visualization. These days, we find quite a lot on the biological scale also. So when it comes to atoms and molecules, but there's surprisingly little, or maybe it's not surprising, in the intermediate scales. And there are quite a few of those. So obviously, here is a big opportunity for future work. It may also be due to the fact that we, in fact, have less data and less basis for visualization available on those scales. And that this is also one reason for why we don't see more works on the so-called meso scales. Another observation that I really found very interesting is, and it's maybe neither a big surprise, is that spatial scales and temporal scales seem to be related quite a lot. Not always, but in many cases, if you are in a small scale in time, you are also considering a small scale in space and vice versa. So if we here have this kind of 2D histogram of visualization publications that we found mapped out across temporal scales on X and spatial scales on Y, you can see, if you want to, that most works are found somewhere here on the main diagonal that signifies a relation between temporal and spatial scales. It's not a perfect correlation, so we do have processes where we have longer time scales and smaller spatial scales and vice versa. But in general, there seems to be a correlation between space and time when it comes to physiology. I guess that physics has a big word to say in explaining this as well. So let's talk about what visualization can actually do, or what kind of approaches we deal with in visualization. And here it already becomes a little ambiguous. So multiscale visualization is easily said, but if you look into the literature, you will see that it refers to quite a few different things. And in the following, I will try to somehow give you a few examples for different types of aspects that we may refer to it with multiscale visualization. Here's just a short first overview of that. So one typical case when we talk about multiscale visualization is a rather technical perspective, where we try to come up with some smart software and or hardware solution to deal with large scale problems. A typical example would be maybe large scale terrain visualization. A couple of years back, this really was a big question. I don't know if you want to have a highly detailed terrain visualization for maybe a flight simulation or you name it, how can you actually achieve that? And techniques were needed that take the viewpoint into consideration, that have levels of detail incorporated, that did smart exploitation of maybe the GPU or whatnot. So this is one category of aspects in multiscale visualization. Another interesting problem is when you actually have data or input from different scales. Sometimes you want to relate some imaging from a small scale to some imaging on a larger scale. I'll show you just one example from the multiscale visualization project in the UK, where they show how CT at different scale levels can be related to each other. So this would be a regular anatomy scale CT combined with a micro CT and even a nano CT, where you have different data sets representing different scales of the same structure. Here it's a bone structure that we are looking at. And there are lots of interesting problems to solve, starting with registration questions and interaction questions and whatnot. A third very important aspect of multiscale visualization is multiscale phenomena. So that's even before we think about the representation in form of maybe data or models. We think that the phenomenon in itself is multiscale, has a multiscale structure or character. Here I'm just showing one example that is quite famous, I guess, in this category, and it's the one of turbulent flow, where we know that we have an energy cascade through the turbulence from large scale structures to small scale structures. And if we want to make meaningful pictures of turbulence in flow visualization, this is something that may be worthwhile to work out. Here's a nice example from a publication of the group in Munich, Rüdiger Westermann and colleagues, where they show two different scales in a turbulent flow. So in gray, you see a larger scale, and then in red, you can see a more detailed, smaller scale level of this turbulence structure in this flow here. And fourth on this slide, I want to add that multiscale visualization can certainly also be seen as a certain type of perspective. It is on the user side, it's a choice of what level of detail you want to consider when you look at some data and when you want to understand some data. And it may very well be that there are relevant pieces of information in your data or model that come at different levels of detail. And it's not at the least the choice of the user to decide for the task at hand which level of detail is relevant, or maybe there are several of them. So in the following, I just want to make a very quick run through a few examples. I consider them as pointers to some relevant work in my eyes. This is certainly not complete. This would take all too much time. And I just want to provide a rough image for you where you could start reading if you were interested in the state of the art. So that's not complete. It's just a few selected examples. I hope you will bear with me. So to begin, I want to come back to this point of smart technical solutions for large-scale biomedical data. And there are many examples, obviously, in biomedicine. We like to image biomedical situations, and with every year we get better technology to have more data. So it's clearly a situation that we are concerned with. Here I'm just showing you examples from some typical applications. Digital pathology, I think, is one clear example that we often have to deal with large images, medical images, biomedical images, microscopy, and also molecular modeling. So a unifying question in a way for this category could be, so there's so much data, now we need some smart technology to visualize it. And Let me just show you a few examples. Here I show you work from CMIV and others in Sweden, Linköping, Norrköping. Klaus Lundström, he will also give a presentation at this spring school later, Anders Innerman and others. And they have early pioneered work in how to visualize large-scale pathology imaging data. Here is an example for 3D pathology data, and you can see that the data set is impressively large with many thousands of pixels in each direction and even a third dimension. And it's certainly not easy to make this interactive in visualization, and in this work they show how they were still able to do so. Here I show you one example from the work of Markus Hartwiger and colleagues, also Hans-Peter Pfister from Harvard and Johanna Bayer. They have come out with a number of very interesting papers in the last few years, where they presented lots of different smart ideas how to enable large-scale data. Here we are talking about streaming data on the pettascale, so that's really large, from microscopy, and with some smart memory layouts and according algorithms, they are able to provide interactive visualization of that kind of data. So this is one example. Another example from a similar authors is to avoid unnecessary computations as much as possible when you have to deal with such large data. So here we are concerned with scanning electron microscopy data that is like 20,000 by 20,000 in size, and then there are several thousand of these images on top of each other. So this is a terabyte-sized data to visualize, and you need to have really good algorithms for that. Here we are talking about raycasting for volume visualization, and in raycasting, for example, it is really good if you can avoid doing computations for 3D locations that do not contribute to the image. So if you have some sparse data, like here, where you have these dendrons and linear structures, quite a bit of the 3D space is not contributing to the final image. So you better don't spend computations on it. And with this empty space skipping technique, sparse leap here, they avoid most of it, and that's what makes it possible for them to have fast visualization of such huge data. Another work that is very impressive is the one of Ivan Viola and colleagues, where they have developed a software called CellView that enables them to interactively visualize really large molecular models. So here we are not talking about sampled data, but geometric models, but highly detailed 3D geometric models. And with CellView, they are capable of showing immense such structures in an interactive fashion. And this has become quite a well-known solution in the recent years. Actually, this work goes beyond the rendering of such structures. They have also devised techniques to actually come up with models according to some generative plans, where they devise, you could think of a recipe for populating such dense 3D molecular scenes based on some principal ideas. So in a way, this is already a work that combines both modeling and visualization on a really large scale. Next, I want to touch upon another important topic in multiscale biological and biomedical visualization, and that is the one of very long time series data. We have mostly encountered this when working with molecular dynamics simulation data, and I'm confident that you will hear about this quite a few times during this spring school, there it is quite natural to really get very long time series out of the simulation. In fact, I guess that at this moment, the limiting factors are not what we can compute, but what we can analyze. In principle, if you wanted to map a molecular process on the nanosecond scale for any significant stretch of a process, then you easily would get thousands to millions of time steps. And I guess we are long from knowing sufficiently good solutions for how to deal with such extremely long time series data. Here I can just show you a few approaches that people have tried to come up with. Here is one from Robert Kincaid called Signal Lens, where he suggests an interactive focus plus context approach to investigate long time series data at different scales. And then one question is usually how to locate the places, the times of interest in your time series data. And the second question is then how to meaningfully visualize it, also in the context of the entire time scale. Here I show you work from Jan Büschke and Thomas Trautner and colleagues. You will recognize a few other names on this paper, where we are finally now talking about molecular dynamics simulations. And here it's not only that you have this very long time series, but you also have them together with spatial information. Often it's actually a 3D simulation over time that gives rise to these long time series. And then after all, you're not only interested in identifying the right locations in your time series, but you also want to connect this to the 3D visualization that has been computed for this time series. And in this work, they try to answer all of these questions to at least a good degree, how to extract the relevant locations in your time series, and then how to derive appropriate focus plus context visualization. What is interesting here is also that this is not only in space, but also in time. So you can also think about how to distort time, even if you're doing a 3D visualization when doing this kind of focus plus context approach. I also wanted to mention work of Perrepot and colleagues. They I've had some very interesting discussions with them about long time series visualization in biomedicine, but not only in this domain. And I think that there is interesting new work to be expected also in the future about this very relevant problem. I also wanted to show this work, even though it's a little on the side of the topic of the spring school, maybe. It's very recent publication in TVCG about long time series. Here it's biologging data. I think the starting point for them are the migration paths of animals, as far as I understood. But they show that they can use this also in different contexts. And here the approach is that a segmentation is performed on multiple different levels, scale levels, in order to boil down the visualization to what can be shown actually in the space and time that the visualization has available on screen. And they come up with a visual query language that they can parameterize through the interface in order to adjust the segmentation at the different scale levels across the time series. And that leads then to a hierarchical approach to boil down from the largest scale to the smallest scales along time. And they also have data which comes with some spatial information along with it. For example, when you have trajectories, then you also have locations, and they provide additional views once they are on the right level of detail to show those. So this were two of very common aspects of multiscale biomedical visualization. Next, I want to go maybe one level more abstract in the discussion and talk about multiscale abstraction. And in the different ways of how to think about this, one is that what I see depends on my observation scale. It depends on what I pay attention to. Here I've just tried to illustrate this famous example of what's the dimensionality of wool. Well, if I look at some balls of wool, I may say, well, they look three-dimensional to me. If I look at a single strand of wool, maybe it's one-dimensional. And if I look at a knitwear made out of wool, maybe I think of it as something two-dimensional. So it depends on my perspective. It's all the same wool, or maybe not the same wool, but it's all wool. But the right scale of discussing it or understanding it or also working with it depends on my perspective. And that leads to this notion of visual abstraction, where I need to choose the right level of abstraction for visualization. And here I show you just a few examples of works that fall, at least in my eyes, into that category. One that I really found very nice is also from Ivan Viola and colleagues, where they thought about optimizing color maps when doing multi-scale visualization. And this leads to an interesting technique where, depending on which scale you are currently at, you are trying to get the best out of the possible color mappings that could be chosen. Maybe I can just show a little video here as an example to illustrate what I mean. So we start, I hope, in a moment with a large scale view onto a populated 3D molecular environment. And maybe I can just stop this for a moment. So on the larger scale, we have a color map that distinguishes different major regions in this environment: the extracellular environment, the membrane, the intercellular environment, and so on and so forth. And the colors are basically used to distinguish between those environments. If we now zoom in, then we basically don't need all of these categories at the smaller scales because they are not visible. So what happens in this technique is that the color map smoothly adapts to the scale level and provides the right distinction between elements scale by scale. So, and it's done in a very smart way that you are not seeing any jumps such that you would get confused by the change of colors, but it's done in a subtle fashion that allows you to still follow the meaning of the colors. So here, for example, we have now, we still have the impression we get the same distinction of the parts of the environment, but the colors adapt locally in order to provide more color contrast depending on which scale we are at. If I go back to this slide here, you can see side-by-side images, and you can see that they are able to interpolate the color schemes from such a segmentation type of color scheme to maybe a color scheme that would be expected for the atomic model scale. Another question is, of course, the geometric representation. Maybe it's usually one question that even gets asked before the color mapping. And here it also boils down to the question, what do we actually want to look at or maybe interact with? In this work by Hauser, he was a PhD student of Ivan Viola and colleagues as well. They represented atomic and molecular models of DNA nanostructures at 10 different scale levels, which they were also able to smoothly interpolate. And you can see that if you were focused on the atomic scale, they had scales to represent the atoms, they had scales to represent the nucleotides and sequences, entire strands, up to the major overall geometry of such a DNA strand. And depending on which level you wanted to consider, maybe when zooming in, when zooming out, or also when interacting with the model, you could choose the different scales and smoothly interpolate between them. That actually led to a work where they created a two-dimensional space that related not only the 10 different scales across each other, but also different dimensionality in which these scales could be represented. So here we see on the left a 3D representation, and on the right we see a 1D representation that is much more abstract, and this can be related to each other across the different scales and the dimensions. So here you have an overview of this map. On the very left, you have the most detailed model. On the right, you have the most abstracted one. And then you have representations in 1D, 2D, and 3D. And that leads to a 2D space in which you can also interact. And here you see a sample path that has been taken through this 2D map, starting in a 1D visualization at a larger abstraction level, moving over to a 3D visualization on a lower abstraction level. And this can be useful, for example, when you want to have interaction. It's often much easier and more straightforward to do some selection in 2D than it is in 3D, for example. So you could smoothly transition from a 3D view to a 2D view, do some interaction there, and then move back, for example. A similar idea has been realized by colleagues of Markus Hadwiger and Hans-Peter Pfister and others, where they are also considering a smooth interpolation space for abstractions. And in this work, I think it's interesting to see that they are interested in small scale features in the brain, in particular, the relation between neurites and astrocytes. And they construct a 2D abstraction space for both of these structures where they can independently abstract each one of them or both together. And they even have a corner in the upper right here where they can go from 3D to 2D in order to get even more abstract and have an overview and a place for interaction. Here, this becomes then a graph visualization as an abstraction from the 3D scenario with the interplay between the neurites and the astrocytes. I think very interesting is very recent work, again, by Ivan Viola in collaboration with Tobias Isenberg and others. Meister Kröller is also among the authors here, where there have been yet another instance of their principal idea of being inspired by artistic illustration. So here on the very left, I've taken a figure from a Nature Review article that illustrates the different scales of DNA structures in a very nice fashion. And clearly, this is a handmade illustration, which has been done very well. Now, our colleagues are able to come up with an interactive illustrative visualization that borrows some of these principal ideas, for example, piecewise linear abstraction structures that are phases where the scales change. And it's now possible with this solution to have a similarly set up multi-scale illustration, but it's interactive. So you can now change what you look at, and the illustration will interactively adapt. So this is a really impressive, very recent work on multiscale visualization and illustration. So next, I want to just briefly come back to this question of features, multiscale features in multiscale biomedical visualization. And it's a question of analysis, and this alone would deserve not just one additional lecture. I mean, here we could go into signal processing, into pattern recognition, and there are loads and loads of work about multi-scale approaches to make sense out of data. The principal idea is that you need to choose a perspective in a way. So if we look at this set of data over the x-axis here, you could ask, how many mountains do you see here? And there is no right answer. You could say, well, I see 2 mountains, I see 3, I see 4, and every answer is in a way correct. It is a question of choice. What is the relevant scale for feature extraction? And I just want to use this example of turbulence here as one example to illustrate this. Some years back, we worked on energy scale feature extraction for flow visualization. So we thought, well, if a turbulent flow is coming with this hierarchy of energy scales, maybe we should work energy scale aware upfront. So instead of a pipeline that does feature detection on the smallest scale, the scale of the data, so to say, and later makes an effort to maybe thin out the set of features in order to make the result manageable. You could try to work on the right energy scale upfront and do the feature detection on the right scale. And in order to do so, one could first employ a method to actually decompose your data into the different scales that it contains. We use principle orthogonal decomposition for this work. And then you can work with feature detection on these scales. Here you can see, for example, if we use this finite time Lyapunov exponent feature extractor on the original data, it gives us some response that we can try to interpret. If we use the same feature detector on a higher scale of the energy cascade, then we can see some of the relevant structures, these vertical structures that are partly responsible for the energy transition between the different scales. We can see them much better in this data. And I've just shown another work that is a little more recent from CGF, where they showed that they can achieve very meaningful visualizations when distinguishing between these different scales in turbulence. Here they are actually suggesting a solution that can appreciate a continuum of scales between large and small scale. You can see a picture of the input from direct numerical simulation. So that's highly detailed numerical simulation. And you can immediately see, you can see from the picture that it's probably composed of different scales. And in fact, if you decompose this into the different scales, you can see some of the big structures, these blobs that are obviously there. You can see them in the larger scales. And then you can, if you look into the details, you can find them in the smaller scales. And they can apply this also to 3D visualization. And here you have a boundary layer of a turbulent flow. And you can very nicely see the different structures at the different scales in this turbulent flow here. And it's quite obvious that, I mean, you could always look at the smallest scale and just that. But if you want to understand the phenomenon on its different scales, you can have a visualization technique that accounts for that. Well, one could ask if I need to choose a scale in order to get a meaningful answer, why not choose them all? Why not work with all the scales instead of just one? That's a valid point, and that leads to this notion of scale space. And is it something that is found across many domains? It's by no means special for biomedicine, but it's an idea where you extend your representation, maybe your spatial or your temporal or your spatiotemporal representation by an additional scale axis. And here I've just show you two pictures from a paper that is very dear to me. It's quite old already, but it's a big inspiration. We have done lots of work on the basis of it ever since. The mode tree, which shows us that if you have data, you can reconstruct it at different scales, for example, by kernel density estimation. And depending on which scale you're choosing, you get either something very smooth without details, or you get something with a lot of details. And then you can come up with a 2D diagram where your vertical axis becomes the scale axis. And then you can basically read off that depending on which scale you use, you have different numbers of maxima in this graph here. If your scale is way too large, then you will always end up with just one maximum. Here you can see that maybe the scale of, say, h equals 1 here is one that has something to say, because for quite a stretch of scales, we have 3 maxima. It's this one, it's this one, and it's this one. But you may also have 4 or 5, and in this diagram, you can actually see them all. And this can then be used for visualization. Here I can show you just one example from Hans-Christian Hegel and colleagues' work, where they have looked at time series data, and they do feature extraction at all scales, and then come up with a visual representation that integrates all of them into one visualization. So here the vertical axis again is the scale axis, and the horizontal axis is time. And you can see that different types of features, they pop out at different scales. We have actually worked on something similar a few years ago, well, 2 years ago or something, where we tried to add a scale axis not only to one of the domain axes, but to space and time. In principle, if you had 3D plus time as your domain and you would construct a scale space on top of each of these axes, you would end up with an 8-dimensional space. And that is a huge challenge, not only in terms of cognition, but also in terms of computation. So in order to manage this complexity, what we did here is we boiled down the spatial information in the data from 3D to 1D, and then we constructed a scale axis on top of each the spatial aspect of the data and the temporal aspect of the data, and use this to study molecular dynamics simulation data. And we could see that we were able to identify some relevant changes in the simulation. Here we were in particular looking at ligands entering and navigating tunnels in proteins. And we found that we could see important differences in this development that wasn't able to see without the scale space approach. So since we know that phenomena of interest are on multiple scales, it can very well be a good idea to actually represent those scales then also. So time is short, and I cannot talk too much about a lot of other approaches. I just wanted to show that there is certainly much more. I want to provide you just with 2 more examples here to give you an idea that multiscale biomedical visualization is way more than what I've just addressed. So the first work is on multiscale visual drill down for ensembles of multibody protein complexes. So here we have a scale axis also across the represented phenomenon. So we have one scale where we are talking about an entire ensemble of such possible complexes. Then we have another scale where we have one of these complexes with multiple interactions between individual proteins. And then we have another scale where we look at an individual pair of proteins. And there we have many atoms that interact with each other and amino acids that are related to each other on the contact zone. So also here we can have, or we have a multiscale problem where the scale axis is across the represented phenomenon. And linking those to each other is an interesting challenge that we worked on a few years ago. And last, just one picture for a big topic that is dear to me, but I can't cover in this short lecture, and that is multidimensional data visualization. So in particular, when we are concerned with numerical simulation, often we compute a whole lot of different attributes for our data. Sometimes also when we have or measurement data, I mean, for example, in cohort study data, it's not uncommon that we have hundreds of different dimensions in our data. And then you have, in a way, you have a multi-scale characteristic in terms of the dimensionality of the problem. In recent work that just came out in PVCG, we have worked on an approach where we construct a hierarchy of dimensional bundles that allows us to have a structured approach to this multidimensional data, where we can say we can bring in some prior knowledge, for example, or we can bring in our tasks in order to steer the dimension reduction and the consideration of different aspects of the data in this high dimensional data, and not do it all at once with just one dimension reduction. There's not enough time to go into details here, but I think that taking a structured approach to heterogeneous high dimensional data is really one of the opportunities that we have in visualization as compared, say, to machine learning. So to wrap up, because time is also up very soon, I tried to give you an idea that with multiscale, we really can mean a lot of different things in biomedical visualization. We can mean large-scale data challenges where we need some super good technology in both in terms of software and hardware to actually make it possible to look at large data scenarios. Another aspect that I think is highly interesting is the one of abstraction, where we ask ourselves how to represent, how to correctly represent a phenomenon such that we can learn the right things from it and the according interactions. And this goes across all the different questions of visual mapping and interaction design. Also there, we can think and we should think multi-scale. I hope that I also could mention briefly that the question of the data and models that we are concerned with is quite important. And sometimes we simply do not have the basis for visualization. So even if we wanted to have some really good mesoscale visualization, it may be not doable because of the lack of data, for example. And then if we go into actually more an analytic situation where we want to extract meaningful features from data, this is an own world of many techniques where we have, for example, questions like reconstruction, multiscale reconstruction, maybe by wavelets or in the Fourier domain or in many other approaches where we maybe do density estimation, kernel density estimation, or the like, and many other more image pyramids. I mean, this is a never-ending story in itself. So to conclude at this moment, I just want to share a few lessons learned. These are just from my side, and maybe they are interesting for you as well. One is that I think the multiscale aspect of biomedical visualization is a lot in the eye of the beholder. It is the it relates to the questions we are asking. And it's not so that there is a right scale in an objective fashion. You can find techniques, I don't know, the Silverman rule for kernel density estimation, for example, that suggest the one and only right scale for your problem. But I think it really depends a lot on the task that you're trying to solve. Another lesson learned is that if we talk multiscale, we should distinguish between bridging a few scales and bridging many. If you want to scale up by a factor of 2 or 5 or maybe even 10, this is in a way also multiscale, but it's much easier than if you wanted to bridge, say, 5 different orders or 10 different orders. And if we go from the body scale down to the molecular scales, we have like dozens of scales to bridge. And this is very challenging in many different aspects in terms of modeling, in terms of computation, and in terms of visualization. And it's not clear to which degree this will be ever fully possible. So that's a big, big challenge, and it's very interesting to see where are the boundaries of what we can achieve. I myself, I find quite interesting to see that there appears to be something like the meso gap in biomedicine, that we are quite good by now on visualizing anatomic structures on the body scale, organ scale maybe, and we are also quite good at visualizing molecules, atoms, but we are not so much working on the mesoscale in between, and this I think would be great for the future. I also think it's quite interesting to consider that our knowledge crystallization process that we often want to support with visualization is multiscale in itself. It has this abstraction in it, and in its nature, this is also multiscale. I think, given those big challenges, I think that we need teams to work on multiscale solutions. It could be that it's just too much to ask for an individual to come up with a really good multiscale solution that answers all of these questions. So it may very well be that if we want to achieve something great in multiscale biomedical visualization, we need teams. And we see big projects like the virtual liver or the virtual brain project, and these are big consortia where people with different professions work together in order to achieve the multiscale solutions that we apparently want to have. So I guess that teams are good to have when it comes to multiscale biomedical visualization. And yes, I mean, I think you hopefully got the impression that there's so much more one could talk about, but I don't have the time for now. So with that, let me just thank you very much for bearing with me, and I will try to see whether you have questions. Maybe there are questions now, or maybe I can look them up in Discord. I'll try and see where your questions come up. Ingrid, are there questions now live? Or
at first, thanks a lot for this interesting talk. So there are people applauding. There was one question that had been removed over your talk because you already answered it. There's one question which says, do you have experience about how often domain experts are the new, do you have experience about how open domain experience are to new visualization techniques?
I have been working with biomedicine here in Bergen now for, I don't know, 15 years almost by now. We first had this MedVis initiative over more than 10 years, and now we have the Mon Medical Imaging and Visualization Center. And there I really see a great interest in the collaboration. One of my colleagues, Arvid Lunderwold, for example, he has always had this perspective on multiscale relations. One example is the brain-gut axis, for example, how to understand how the brain and the rest of the body are working together. And it seems to be clear that we need to take a multi-scale perspective onto that problem. And they are very much welcoming the cooperation with visualization. So I at this moment, I have the impression that it's nice and straightforward to collaborate with the domain. And I just would like to encourage this to everyone. I think it has been becoming maybe even easier than it was some 20 years ago.
So there's one more question or
Yes.
comment saying, isn't mesoscale always there in the gaps between all the scales that we have worked on?
Yeah, it is there,
How do you know when we are done?
Well, I mean, it's there, but the question is, can we get a hold of it? Can we get our hands onto it? So obviously, it is there. I mean, if we think that between the brain activity, say the cognition that we are doing, hopefully, and it's the signaling between the cells, there is the whole cascade of scales. But do we have the data for the mesoscale? Do we have the models? Can we relate them to each other? Or is it in the end something that is a little disconnected, where we have one picture that focuses on the molecular scale or the atomic scale, and another one that focuses on the large scale, say the network of brain activities? I think at this moment we are often not connecting the largest and the smallest scales, and that's what I try to indicate with this mesoscale gap.
Okay, so far I don't see any more questions here. Maybe there will be more that you can find on Discord. Thanks again for the interesting talk. We all have a lot to do, to work on in the future now.
I agree.
I agree. Yeah.
Thank you very much.